from Microbe TV. This is TWiEVO, This Week in Evolution, episode 112, recorded on the 26th of March, 2025. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. And I'm using a really good microphone, so you like me more. Also joining me from Salt Lake City, Nels LD.
Hey, Vincent, good to be with you. Stumbling out of the blocks here a little bit. I think hopefully my high quality audio is coming through. I'm a little choppy on my video stream, but I think that's been happening for some reason on my computer. Hopefully it will pick up
Nels,
in a moment here.
Your brain makes up for any deficiencies in video and audio.
Yeah, right. We'll put that to the test. We also put our listeners to the test here. Thank you for your patience, everyone. I think we're coming in a couple minutes late. It's a couple of things. First, it's a pop quiz. We're seeing who's really devoted to our TWiEVO broadcast, who will hang with us through the technical difficulties. So thank you all for your patience and for being here. And secondly, we're just, Vincent, I think we're just modeling how scientists work. We're usually, a couple minutes behind, a little disorganized, just figuring out the direction we're going, flying by the seat of our pants.
Yeah, you have to just flow with it.
That's right.
And that's what we're doing. Tell us where you're from. We'll say hi before we get going here. Got some interesting science for you today. Mm-hmm. So if you've already introduced yourself, the first chat I hear here is from Jan T. So if you've said hello before that, re-say hello. Jan's from Tucson, 90F. Your temperature over there, Nels?
Yeah, we're warm here, not 90F going up that high, but I think we're going to approach 70, 75 degrees on the Fahrenheit scale. So yeah, it's unseasonably warm, but with, snow on the menu probably by this weekend. So we're kind of in the freeze thaw zone. What's it looking like out on the East Coast?
Here it's cloudy and 9 degrees Celsius. It's a bit chilly. I'm still wearing a hat, wool hat and gloves, and a big puffy coat. It's too bloody cold. By the way, I want to thank our moderators for being here today. We really appreciate you, Barb Mac
Yes.
UK, Vanity Nutrition, and Tom Steinberg. Thank you so much for moderating. We appreciate the time you spend here. In exchange, we offer you knowledge. That's what we can do, Nels, right?
Doing our best, combing the science literature, broadly speaking, and finding the best stuff that we think is the interesting stuff related to evolution. And great to be together in conversation as a community.
Tom is in Oregon, and he says, intriguing, dense paper. Glad to see the University of Oregon work here. Of course, that's where Tom is today, in Eugene. Cool.
That's right. It's all woven together. So we'll get into the details here, but this is a local one for you, Tom.
Lise is in Columbus, Ohio. Welcome to the stream. Of course, John is in Eden Prairie, Minnesota, 49F. Do you know where Eden Prairie is, Nels?
Of course. Yeah, we used to play Eden Prairie in basketball when I was in high school. Eden Prairie High School, had some buddies there, one of the suburban zones around Minneapolis, the Tri-Metro area.
John says, try not to leak any DOD secrets to the mods.
Yeah, I think we have a lot of ground to cover as it relates to the federal government as we're thinking about it directly, and we'll highlight some of that as it relates to science, some updates there. But my goodness, it feels like we're seeing the same patterns across the board here. I don't know, Vincent, it feels like, it's been about a month since we've been together. It feels like about a year or a decade in terms of all of the things that have happened.
A lot of things have happened, a lot of unfortunate things. I don't know, has anything good happened?
Yeah, that's a fair question. We should flip it around. I think I am seeing a few good things. So one of the events that I was involved with a few weeks ago was the Stand Up for Science. We had a local sort of chapter that emerged. So this is hopeful. So this was organized nationally by 5 grad students. And I don't think I can do justice to all the details right here, but so grassroots effort. There was a recent graduate in our program here at Utah who took up the mantle to organize one here at Utah. We got out on the south steps of the state capitol. I'd say more than 1,000 people showed up on short notice on a Friday afternoon. And yeah, we got together as a community to say, hold on, here's why science in our backyard is so important, so valuable, and came together to make the case, but also to celebrate that with each other. And I think there's definitely some hope in that. I'm kind of thinking about the local levels, the sort of grassroots or the little shoots as hopeful sort of signs, because yeah, on the national level, things are pretty bleak to say the least.
I think, as we've said before, many people do not appreciate the value that science brings to their lives. So we have to reinforce that, because, they're looking at the cuts and they go, okay. They're telling us it's wasted money, so I believe them. But put it in context. Like yesterday, people who have COVID grants got an email from NIH saying, we don't need this anymore. Pandemic's over. But that's not why you work on COVID. We continue to work on influenza and there's no pandemic because we want to understand it. We want to be better prepared for the next one. There's a singular lack of understanding of why you do science research. And you cannot break the system and expect that it will come back very quickly. This is very, very bad what they're doing. You have a lab which is paid by grants. You take the grants away, everybody's gone. Everyone who's trained to do the experiments that you need to do, they're gone.
Exactly. Yeah, this notion that it's sort of like flipping a light switch on and off is complete nonsense. So I spoke at our local Stand Up for Science event, Vincent, and one of the points I made is that in my lifetime, I haven't seen sort of as big of a disconnect in the government between confidence and competence. I see a lot of confidence that they're doing all the right things, but no competence underneath it. And that disconnect is leading to an absolute collapse. That the AVID grants that you just described, they're not just working on SARS-CoV-2. The treatments that were being sort of fast-tracked or really turbocharged with those resources, or thinking about all kinds of other viruses, including potentially SARS-3, SARS-4. We don't have to look back too far in our time to see some near misses, whether it was SARS-1, MERS, Middle Eastern Respiratory Syndrome, and then SARS-2. And so, this notion that as it's sold both on sort of multiple fronts here, it is really a shame. And I think the thing is, it's getting where you can see this sort of in all facets of government as the cabinet is starting to, come into force and exercise their confidence with low levels of competence, as we've seen, obviously, in the headlines, even as we're speaking right now, as it relates to how the U.S. military is conducting operation. It's just, I think you scratch beneath the surface right now at any governmental agency, and you have these kind of both external targeted attacks all the way down to sort of this more quiet dismantling that's going on as it relates to funding. So in addition to these kind of headline grabbers, Vincent, I was just looking, so Jeremy Berg, who's I think I've brought up in our past couple podcasts, who's a former head of NIGMS, one of the great national institutes of health, has a dashboard tracking how funds, NIH funds earmarked, agreed to by law, by the Congress, has been starting to distribute or not distribute funds. And so if we look at last year's distributions, up till from sort of January 1st to today, there was about $4 billion of non-competing grant funds that were released. So that means you already have a grant, you've gotten through peer review, this sort of commitment from the NIH for 4 years, 5 years of funding. It might be in year 1 or year 2. So that's non-competing. It's just meant to go into the next fiscal year. We're $2 billion plus behind on those distributions compared to last year. And similarly for competing grants. So this is you've gone through peer review, your grant scored well. In some cases, it was the top 10%, or the top 1 in 4. In other cases, depending on sort of the study section, it's gone through sort of the gauntlet. And then right as it's ready for funding, It's just disconnected. So we're 2 billion down there as well. And so that sort of eroding, that erosion or that dismantling is a symptom of the bigger disease here. In addition to these headline grabbers where they're taking away whole programs, we've seen some, I think really awful scenarios at Columbia University in terms of the 250 million that appears to be in jeopardy, for 400 million across all departments, 250 million specific to NIH, and then another 175 million at Penn. And what are the reasons for this? It's just the notion that this is how we run science funding is just is abhorrent. We've been, I've been, of course, then at the local level, what do? You start to talk to your representatives. We've been doing that here with our junior senator, John Curtis, talking to some folks in his office. And they start to express this as sort of, well, it seems like the scientists are feeling a little anxiety, or but this isn't just anxiety. This is anguish when you see this incredible system just getting dismantled at various levels. It's really awful.
You cannot just do this. If you want to change some direction, there's a way to do it properly. You tell people, when your grant is over, we're not going to renew it in 3 or 4 years. And you give people time to refocus and do other things. You don't just cut it off. Cutting it off shows vindictiveness. It doesn't show any logic whatsoever. And so you're destroying the science establishment. And you're not even thinking about that because you need science in this country to survive. You have 10 years of no NIH science, you see what happens. We're going to be in big trouble, not just infectious diseases, but all kinds of other things as well.
I agree. And it's, the case we're trying to make is that it's not sort of like a jobs program for scientists or STEM majors or something like that. This is taxpayers' money. These are the treatments, the cures, the interventions of the future to promote public health. That's all of us. That's all, so it's not Democrats or Republicans that get cancer. It's everyone who gets cancer. It's not Democrats or Republicans that get influenza or SARS-2 or measles. It's everyone who gets it. And so, that sort of that breach of trust for what scientists beyond sort of just the career opportunities, et cetera. I mean, it's really we're it's being exposed slowly but surely. Unfortunately, hopefully, not the pacing here is pretty critical. It seems like to try to get some kind of collective together for countermeasures, and what that looks like is a whole nother conversation. But this isn't just sort of this is serious, as we're seeing other serious events as it relates to military action, as it relates to Social Security, the investments there,
Medicare, Medicaid,
correct, all of the bedrock. And then when you put people in charge who just aren't up for the job or have a very sort of narrow agenda in terms of their own self-interests, things go off the rails really badly. It's wild how it really exposes how vulnerable I think these systems are, and how much trust we depend on each other for these things to sustain.
Yeah, I didn't realize they were so vulnerable, but obviously they depend on people working together in government, which right now they're not. They're just blindly following someone who,
that's right.
I don't know what he's promised them, but if they thought about it, they would be appalled, and they are not. So, it's just pathetic. It's just pathetic. But we can say all these words, but it's still going on, and it's going to be a real problem, folks. You better tell your friends that without science research, we're going to be in big trouble.
Yeah. No, it's a mess. And the sort of the definition of efficiency here is doing a lot of work to cover up a lot of things that are very much not efficient, and certainly in the longer run.
Oh, they also say, we're going to focus on chronic diseases. Okay, that's fine, but you cannot exclude other kinds of diseases because they're real. And if you ignore them for 5 years, you get in big trouble. And I should tell you that many viruses cause chronic diseases as well. So to make that kind of divide is just dumb. I don't think there's anything realistic about this idea to focus on chronic diseases. People have been working on chronic diseases for years. It's very difficult. You're not going to make a difference I don't think this money's going there, Nels. I think it's going to go in the pockets of very wealthy people once they get their tax break.
Yeah, no, for sure. And I mean, part of the continuing resolution that passed also has some bad news for in terms of impoundment and what that, so where are those, what you're saying, Vincent, where these funds are going, how they're being distributed. So, I think we're all waiting for the next shoe to drop among many dropping shoes here. One of them is the new director of NIH, which is this health economist called Jay Bhattacharya from Stanford, who had some really wild ideas related to how we should contend with the SARS-2 pandemic in terms of, before a vaccine existed, and how what lockdowns or not doing or letting it run sort of buck wild would look like. And so, how much of this is top down? It's in top down above him. So with RFK Jr. now commissioning sort of an anti-vaxologist to do a new study on autism links. I mean, this just, at some point, we're just kind of pretending to do science as opposed to doing it. And so the more top down it becomes, the more sort of artificial it feels like it becomes. It's one of the beautiful things, amazing things about the NIH and sort of the American style of science is we just compete like cats and dogs and let the best ideas win. We don't always get it right. And I don't think any of us as practicing scientists are saying that it's perfect. And you can find plenty of threads of us complaining about, wow, those peer reviewers really got this one wrong. And it's true, but that's kind of the same way, like when you're playing basketball, you complain about the refs, but you love the game and you keep competing, you keep playing because it's so good. But when, we see action like this, and as it cuts deeper, it's sort of like, oh, well, we're taking 2 players off your team, but play against the other 5, or, oh yeah, we just took down one of the hoops, but just throw the ball against the wall. It's kind of pretend. It just becomes fake. And it's just, on one level, it's just silly. It's just sort of going through the motions. But on another level, that's really dangerous, as you're pointing out, because if we're pretending to do work as opposed to doing the work, now we're letting up on the gas pedal. We're no longer putting our all into figuring out how biology works, how we can think about things differently in creative ways that can lead to new pathways, that can lead to interventions of the future to mitigate pandemics of the future, to mitigate chronic disease, all of the above. But yeah, it's wild times, to say the least. And
it's
really
fake.
troubling.
If you want to investigate a new area at NIH, you make a program announcement and you make a long document saying we're interested in grant submissions in these areas addressing these problems. I guarantee you that we're never going to see such a thing for chronic diseases. They're not going to do anything. I guarantee you. And if we do, I apologize. But I don't think we're going to do that because I don't think that's really the purpose. It's just to divert the money to something else. Yeah. Oh, dear. It's very frustrating. I'm glad to have you to bounce off, Nels. It's very good.
Oh, no, me too. I really enjoy these discussions, and we're going to be having a lot more of them moving forward as sort of slow motion train wreck continues to unfold. Yeah, it's really wild.
Don't tell me I'm conflicted. Don't ever tell me. I don't do research anymore. I don't get any money from Big Pharma. I am not conflicted. All I do is talk about research, and if there's no more research in the U.S., I'll talk about research in Europe and other countries, okay? So I am not conflicted. So just shut your mouth if you're thinking that Racaniello is conflicted, because that's what RFK Jr. would say. That's his answer to everything.
Oh, right. Yeah, exactly. Like somehow we're standing up for the sort of big vaccine or something like that.
He's the most conflicted person in the world because he's working for law firms that sue vaccine manufacturers. And if you tell him that, he'll say, look at your conflicts of interest, which he did to Bernie Sanders, and Bernie has none. Okay. Yeah. Anyway, let's do science. Let's finish saying hello to people. Gary is from Denver.
Hey, Gary.
Very cool, where I may be this summer. Welcome. I don't know. That would be cool. Yeah. Ooh, yeah. Abdulaziz is in KSA. Welcome.
Welcome back. Good to have you along.
Other Shade is in Amsterdam, where it's 11C. Day's almost over, I guess. Jason, so you got to get off it on a live stream. Yeah, that would be fun. I'm sure that would be fun. But he might not do it because a lot of people like to attack him needlessly.
True. Yeah, we're kind of a couple levels back here in the evolutionary sciences. We're maybe a little less exposed on some of the politics somehow. Although that's a whole nother. The politics and evolution isn't new either, especially as it sort of comes to a confluence with religion. So intelligent design, right, and some of the sort of notions along those lines have been on the menu for a long time in evolutionary biology. I mean, that's one of the things I think is important here. And so for each of us as kind of practicing scientists, when we have the opportunity to stand up to do so. And so, I mean, that's the other kind of maybe I think important storyline that I haven't seen expressed a lot, is that sort of this idea that, oh, well, scientists are kind of wimpy or, trying to hide in the shadows. Scientists are pretty, have done a lot of on-the-job training and resilience, sort of the practice of resilience under sort of uncomfortable or unknown situations. So because the resources aren't always so clear, we compete like cats and dogs for grants and funding and everything else. It's a pretty familiar notion to feel an uncomfortable space. There are tipping points, and if you push across that, the whole thing collapses. But if increasingly, I think we're going to be a country that's facing sort of tipping point or catastrophic moments. And so the good news is there's a whole roster of scientists that are pretty familiar with things getting uncomfortable or getting tough. And so starting to connect scientists to the ability to sort of intervene or just to communicate and to share perspectives, to get into conversation with scientists. I think this is a critical time to do so.
Ivy is in Madeira. Nice.
Welcome.
15C. Emmy Wheeler's in Helsinki, where it's pretty chilly.
Mm-hmm. Welcome back. Good to see you.
Sue is in Coos Bay, Oregon. Nice.
Nice. And nearby where our paper was, where the research was unfolding.
Oh, cool.
Not too
Jason
far.
is in Las Vegas.
Welcome.
Pete is in London.
Welcome, Pete. Good to see you again. Hope the flooring starts to cooperate as we're podcasting here. It's live streaming.
Okay. Thanks for coming, everyone.
Yeah.
Hope you enjoy the science.
We've assembled a good crew, so let's get started, Vincent. The paper today, published I think a month or two ago, is titled Cephalopod Sex Determination in its Ancient Evolutionary Origin. So before we kind of dig into the details here, let's talk about the authors and who did the work. So first author, Gabrielle Coffing, and then a group of researchers, all from Eugene, Oregon. Senior author is Andy Kern. So at the Institute of Ecology and Evolution at the University of Oregon in Eugene. Great place to practice science, great university there. And I've been a little bit remiss, Vincent. I was meant to say this last time, is to kind of highlight what the federal funding of the work that goes into the papers or the studies that we highlight here on TWiEVO. And basically, the vast majority of them depend on federal U.S. funding, including ones that have international sort of ties and collaborations. So this one, for example, there were 4 federal grants at play. So one is a National Science Foundation or NSF graduate fellowship. And then there were 3 NIH grants that helped support this work. One is a so-called R35. That's sort of a kind of a program grant for an individual lab, and 2 R01s. These are the R01s are sort of the workhorse of NIH science, where individual lab PI applies for 5 years of funding. And so, I think the deeper point here, and for a lot of our studies, including this one, probably the majority of them, they don't all sort of rise or fall on just one grant. There's also some more internal funding from the University of Oregon that came into this, some of their genomic sort of facilities and resources that's underlined by some of the indirect costs that come from some of this work or from other work. But it's almost like a tapestry. It's all woven together, all of these resources. And so, if you it's kind of like playing Jenga, you can pull out one piece and maybe the tower will still stand. You can pull out 2 or 3 pieces. It gets more wobbly. You pull out another piece, the whole thing collapses. And so, I won't belabor this at the moment, but this will be kind of a topic on the menu too, is how federal funding sort of makes this possible, or kind of is the scaffolding or the foundation for this kind of work on many levels, and how important that is. And so, and this is one example of that. We could probably, and there will be, science to come for a few years, but it will be the sort of tempo of it and the amount of it will really start on our current trajectories will really start to become curbed. It'll be harder and harder to find sort of viable labs that can sustain these kind of programs over the years it takes to do this sort of high degree difficulty science. And so I want to add that to the conversation going forward as well. NSF funding looks like it could be in real trouble, like 50% level cuts. And then NIH, as we were just discussing, it feels like everything's a bit up in the air. We're massively behind on funding for this year. And then we're looking at potentially some top-down stuff and a lot of political interference as it relates to some of the headline grabbing sort of examples at Columbia and Penn, among others. We've seen it here locally too. So there's a big grant that just went off, that got targeted and got pulled off the table, got terminated 2 weeks ago, involved in clinical and translational science here at Utah. And so this is all, it's just wild, the different levels it's being attacked.
But it wasn't making transgender mice, was it, Nels?
No. But the, I'm wearing my Penn t-shirt today. This is I was glad to see that it sounds like Penn is starting to stand up to some of the attacks that it's under for the $175 million of funding. I'm hoping they stay strong. So this is all predicated on the notion that there was a transgender swimmer 3 years ago who was in the swimming pool at Penn at a meet, and somehow that's a reason to drag down hundreds of millions of dollars of funding. It's just a really, on all levels, just a really, as you were saying, Vincent, a sort of pathetic way to run, for us to try to run science in this country.
Instead of working together, they're just being bullies and being vindictive. What's going on at Columbia is a real problem. Okay, so the president is giving in to the administration demands, and supposedly they will get $400 million in grants back. I don't believe it. I don't believe there'll be others cut in the future, and they'll say, "Oh, we don't like the way you're responding to this. We're going to take this away." It's all blackmail.
Yeah.
So
Correct.
It's a problem because you're also setting a precedent for other universities. So, when you think about it, $400 million in a university the size of Columbia, that's nothing. I'm not sure I would do it in the same position. I mean, many professors are really pissed off that they're giving in.
Oh, right? Yeah. Well, and I think I read somewhere there is an official at the Department of Justice that said that, as they're giving in, like, oh, that's an okay start, but I haven't seen enough for us to reopen the funding. I mean, it's just so, these are sort of bullying tactics. And so exactly. how we organize broadly to stand up to that, I think, is going to be really important. And so that hint that Penn is doing that, I'm celebrating that. That's one of those sort of glimmers of hope, slivers of hope, is that we're organizing, standing up, and trying to counter some of this by sharing what's happening going forward. So, in speaking of sort of a sharp transition here, as we're sort of volleying between our science and our it's all woven together. So, and this is where we are, honestly, right now. So, we're going to talk about the evolution of sex and sex determination, which I think has, maybe we'll close on some of these ideas, circle back to some of what we're hearing as it relates to federal policies about there's only two sexes, et cetera. And so actually thinking about this in terms of putting this into an evolutionary perspective and thinking about the real biology here, I think is actually really important in this moment as well. So sex and sex determination is a fundamental topic in evolutionary biology. Some clear examples of this is there are some critters out there that reproduce asexually, and others that do so sexually. And what is the sort of cost benefit analysis from an evolutionary standpoint of sex? And so if we think about getting our genetic material into the next generation, or genetic material persisting sort of through that Darwinian lens, actually having sex is a huge drag on genetics, right? So you're basically, by partnering, with someone of the opposite sex or of a different mating type, depending on the species in question, 50% of your genetic material is not going through because you've come to this compromise, or you're sharing half of your genetic material. And so why not just clone yourself or divide asexually like a lot of microbial populations do? And it turns out there's probably the best idea out there that I've heard, at least, or one of the major ideas out there, is that you're willing to pay that cost by sort of the benefit of recombination, of putting genetic combinations together, sort of genetic diversity together for the first time in new combinations, that sort of diversifying act sort of is worth it. It's a payment worth making to give up half of your genetic material, if that means you're more likely to survive because you're more equipped to handle a changing environment, or to counter the infectious diseases that can really have a negative impact on fitness. And so this is the Red Queen hypothesis again, was actually sort of brought up through the idea of trying to explain why sex persists. And so asexual reproduction also works, but as the sort of population becomes more genetically homogeneous because you're making near or exact copies, then the notion is that population will become more vulnerable to either catastrophic environmental events or catastrophic sort of pandemic events that repeatedly sweep through.
And of course, you can also limit genetic variability in sexually reproducing populations, right?
Absolutely, yeah.
So like chickens on chicken farms, they're very genetically restricted. Yep. So because they're all made from one breeder. And this is why you cannot just select for H5N1 resistant chickens, because there's not enough genetic susceptibility, differences, and it's 100% lethal. Won't work. Go back to genetics school.
No, totally agree. And this is, in sort of animal breeding, this is inbreeding depression, right? Or where you, or there are some consequences depending on that gene pool. It also is obviously kind of front of the mind for conservation biology as populations crash, and then we try to, intervene in some cases to help counter, the extinction of species. If you have a really limited gene pool, you might, sort of nurse that population along for a while, but it could be really vulnerable given the inbreeding that's sort of necessary to bring it through those sort of situations. So, and we've talked on TWiEVO about sex determination. How do you then? So, if we're committed to sex, or if that works as a sort of strategy for fitness over the long term, how then do these systems develop, or how do they evolve? How are they encoded in genomes? And so this is the topic of sex determination. We've covered this on TWiEVO before, sort of in our early days, Vincent. TWiEVO 35, sex or strawberry sex chromosomes forever.
Strawberries.
So some really interesting and wild work related to how sex is determined in crosses of strawberries. And so here, some of the genes that encode the functions that determine sex actually move across to different chromosomal locations. And so it's this really sort of volatile or unstable or changing situation of how the genetics encoding sex determination happens there. And that's not just strawberries. Like, there's all kinds of really cool work in many different systems, including fruit flies, Drosophila, some of the species there, really young sex chromosomes. And so it's sort of like fixing something that's not broken. How sex determination is, determined, or how sex determination is encoded in our genetics, is not sort of this immutable thing that never changes. There are, and we'll talk about cephalopods here in a moment, there are some cases, and this is one of the oldest sex chromosomes that they both discover and then do some evolutionary analysis to describe. But it's the Wild West out there in terms of the genetics behind sex determination among a lot of species. So some other, I think, kind of exciting examples that are textbook examples are things like turtles and fish, or turtles where the temperature, so the eggs as they're hatching, depending on the temperature at which they're incubating, that can have the environment influences, whether it becomes a male or a female turtle. For fish, depending on the kind of local population you're in, if all of the males clear out or are consumed or gone, then a female is nominated to become a male. So kind of in real time, sex determination can unfold differently depending on the sort of signaling or the communication as the fish in schools are behaving. One of my favorites, kind of really off in the weeds here, is in my PhD critter, this single cell pond critter called Tetrahymena. It's a distant cousin of Paramecium. There's 7 mating types in Tetrahymena, the single cell critter. And so depending on, again, environmental cues, you have this diversification of mating types. And then if you're of mating type 1, you can mate with 6 other mating types. A really interesting expression of there's just this sort of this wild elaborations in sex determination.
I guess that's so you're likely to encounter a proper mate, because maybe it's too rare otherwise if you just had 2, right?
Yeah, exactly. It's not a 50-50 proposition. It's like a 70, whatever, I'm not doing the math right, 80% proposition that you just encountered someone where you're compatible for mating. Okay, so here we pick up the case with cephalopods. So we're talking about octopus, squid, cuttlefish, and company. And kind of
really cool organisms, right? And they
Yeah.
say how they're really smart. And I stopped eating them years ago because they're so smart that they shouldn't be eaten.
Yeah, when octopus comes out, I have to confess, I won't order it, but if someone does, I'll eat it. But I feel a little weird about it because these really are, intelligent creatures.
Well, the best example is this short animated cartoon called Octopody, where there are 2 octopi in a tank of water in a restaurant. And the chef pulls one out and is about to chop it up, and the other one rescues it, and they chase throughout the city. The guy chases them until they finally escape into the ocean at the end. It's just great. It's just great.
I think this is also the plot line in Finding Nemo. Like this is octopus have taken up our sort of artistic imagination for good reasons.
Yeah.
So actually, in terms of evolution or genomics, relatively undersubscribed in terms of the work or what we know about them. So including not really a lot of high quality genomes. So here the authors kind of take up the case by doing really high quality, using sort of recent advances in long read sequencing in particular. They start to take up or to improve the genomes really drastically. And so using both long read sequencing and then sort of new technology to do better full length cDNA or mRNA sequencing. So these are all the genes that are expressed. So as we go from DNA to RNA to protein, you start to get that catalog. This allows you to annotate the genomes better, to assemble them and have a better sense. So the species they picked here is a female or individual is a female 2-spot octopus, where they put a lot of these resources in. And so, it turns out, so it wasn't obvious from cytology. So if you look under you can look under microscopes at chromosomes, and sort of under the right conditions, you can kind of spread them all out. This is in the parlance of genetics is called a karyotype. So 23andMe for humans, we can, you probably have all seen these chromosomal spreads where they're all lined up. The cytologists have arranged them. So here's chromosome 1 that you got from your father, chromosome 1 that you got for your mother. There's 2 of them. And then you keep going down the list. And then if it's a male, there's an X and a Y chromosome sort of at the end of the karyotype. If it's a female, it's XX, 2 X chromosomes in humans. So it turns out in octopus, other cephalopods, you can't do that. You can't. It's hard. Like people have tried this, or they've done karyotypes, but it's not clear that there's a sort of heterogametic set of chromosomes that would be the sex chromosomes. And just to stipulate here, sex chromosomes are again highly diverse. Some of them are ancient. And encode thousands of genes. So it's not just the sex determination genes on there. We call them the sex chromosomes because they include those. But they can also be in Y chromosome case, that can be a kind of a gene desert. And it's actually a really fascinating area of ongoing genetic study is the deterioration of the Y chromosome. So if we come back in a few million years, assuming there are humans around, there probably won't be a Y chromosome. There'll probably be a new basis of sex determination that's encoded in our genome. So again, not immutable. These things can change. And in fact, they can turn over in some species really rapidly, as is being actively researched in Drosophila. Okay, so a big sort of gap in our knowledge, both just in all of genomics for cephalopods. I think there was a There was an old assembly from 2015, about 10 years ago, of the two-spot octopus, but it didn't like reveal some of these sort of chromosome level insights into things like how sex is determined, et cetera, or what are the sex chromosomes. And so here, to kind of preview our sort of take-home message, they find out the female has just one sex chromosome. They infer this from some data that we'll walk through. They call it the Z chromosome. And so the karyotype here for females would be Z0. You just have one Z, and that means it's a female octopus in this case. The males, on the other hand, have two Zs. And you can find this by doing sort of comparisons all the way through other cephalopods. It's probably the case that this has been going on for 500 million years.
ZZ Top.
Oh, there you go. We have a new entry for our title here. We have to come up. I think I had Sex Z cephalopods, but it might be.
It's pretty good.
ZZ,
I like that.
Yeah, it's okay. We'll see where it goes here. Anyway, so again, sort of this flips the script from our own sort of sex chromosomes. So for us, the female is the homogametic XX, whereas here the male is homogametic ZZ. And then for humans, XY, that's a heterogametic situation. Here that's not the case. It's just a single Z, what's called a Z chromosome. There are humans that are XO. This is called Turner's syndrome, and has a number of impacts, not just on sort of sex determination or sort of sex traits, but other things like sort of physical traits and features, in addition to fertility and other things like this. So again, sort of not this immutable thing. It's not just XX or XY. There's other cases where the chromosomal or the karyotype is different. That's really common in human populations. We don't talk about it much, and it's being obviously pushed to the margins or attacked in real ways by the federal government at the moment. But anyway, we'll stay for a moment here on the octopus. And sort of dig in beneath the surface. So how did they pull this off? So they did tons of what's called pack biosequencing. This is long read sequencing, one of the platforms that's emerged, really powerful. You can get so you chop up the genome into small pieces, the chromosomes into smaller pieces, and then you get long reads. So, and by long, we mean, like 10,000 base pairs plus. So each base pair is one of those ATCs, Gs, the letters of the genetic code, our DNA. And then they use another trick here, which is pretty common in modern genomics, is chromosome conformation capture. It's also called Hi-C. And this allows you to sort of get a sense of where those fragments relate to each other. Are they on the same chromosome? Are they right next door? Are they distant on the same chromosome? Or are they on a different chromosome? And sort of putting this together, then you get a chromosome level assembly. They find 30, chromosomes. That is actually consistent with what cytology has been done. But then where they actually can make new progress on the sex chromosome is they notice for one of their chromosomes, it turned out to be number 17 here, the female only had half as many reads compared to males. And so that's how we start to get at that Z0 versus double Z, or ZZ top, as you're saying, Vincent, for the male octopus here. So, when you have an observation like this or a hypothesis in science, you want to kind of test it and try to get more evidence for or against it, I should say. And so they grab a couple of more genomes or octopus, both males and females, and they do sort of short read sequencing. So, looking specifically at the copy numbers as it relates to chromosome 17, and it looks consistent. So if you're a male, now in multiple cases, you have sort of the normal diploid or 2 copies of chromosome 17, whereas if you're a female, you just have 1. And so this state of just having 1 is called being hemizygous instead of diploid. And that's not so unusual, but a little bit different. So birds are another sort of famous example. We call their sex chromosomes Z and W. And here and same for reptiles. So here the females are ZW and males are ZZ. So a little bit different. So the females of birds and reptiles are a little bit closer to us. It's sort of a different basis, but it's also heterogametic in that case versus hemizygous. Okay, so that's sort of, that's the evidence that they put forward for this being the sex chromosome. They do a little bit more sort of later in the paper about what are the genes, sort of candidate genes that might actually encode sort of the genetic basis of this. And they find the presence of some of these genes on the Z chromosomes of octopus. So that's consistent. They also observe a high number of retrotransposon sequences. This is pretty interesting. It's sort of, they don't really pick this up, but kind of one of the fun things about science, right, is you sort of get a new observation, you sort of put it out there to the world, and then someone else might pick that up, see that observation and say, hey, that's pretty interesting that there's all of these sort of selfish gene elements, so-called lines and signs. These are long interspersed nuclear elements, and signs short interspersed nuclear elements that get propagated above Mendelian inheritance. They get overcopied and are showing up as in abundance on these Z chromosomes. They have sort of two times the number of these transposable elements than the autosomes or the other chromosomes that don't encode, aren't involved in directly in sex determination. And then, so then they do some comparisons, some evolutionary comparisons to ask, because chromosome 17 or the Z chromosome, when did it arise? So to do this, you just start sampling modern species. So you ask among squid, among other, do they also have a sort of equivalent Z chromosome? And here they can kind of mine some of the existing literature. And the answer is yes. So it looks like basically, and they did some sequencing themselves, actually, kind of based on what they discovered in 4 other cephalopod species. So now they're getting into cuttlefish and squid and so forth. And they see similar patterns. So basically that, copy number, there's only one evidence for one sex chromosome or Z chromosome in female cephalopods of other species, modern species. And so then when you work back to the inferred common ancestor, that dates the presence of this. We think it's been around for at least 480 million years, something in that ballpark. And that's, turns out, for sex chromosomes, that's maybe approaches the record book for the one that's been sort of the most stable based on these findings so far.
And that's where 480 million years ago is where all of these current day species diverged, right?
Exactly. Yeah. So there were critters that probably looked like some, kind of ancient form of cephalopods, octopus or something like that, squidish creature that probably encoded the same thing. That turns out that's more maybe the rule than the exception. A lot of other sex chromosomes turn over a lot more rapidly. Our Y chromosome in humans is in the process of doing that now. And then neochromosomes show up. So what happens is through processes that are still sort of actively under investigation or learning about, but as an autosome, one of our regular chromosomes somehow picks up the genes that encode sex determination and sort of overwrite the other chromosomes that are out there. And so that gives you so that's been happening repeatedly over evolutionary time, and kind of continues to sort of echo going forward. And so, lots of important kind of take homes here, I would say, or reasons to care about this. So some practical ones, so understanding the basis of sex determination sort of gives us the ability to intervene in ways that could be beneficial. And so I guess for cephalopods, and specifically, I'd give 2 obvious examples. So captive breeding of cephalopods is hard. So there's a lot of octopus, we're both a little squeamish about finding it on our plates on the menu, but around the world is, a sort of a cornerstone of a lot of cuisine. And it's really hard to captive breed octopus. So knowing or getting closer to understanding how sex is determined can really add to the tools available to make this possible. I think there have been some advances recently in captive breeding of octopus, and this could sort of help to promote or to support all of this sort of aquaculture related kind of the agriculture of the sea, basically, or of cephalopods. Calamari, another obvious example there, for sure. I'm less squeamish about calamari. What about you, Vincent?
I try not to.
Oh, wow. Yeah.
They're related to octopus.
Okay.
So.
Wow. Good for you. But, another massive facet here is conservation biology. So as the impact of humans on both the habitats, but also as we domesticate or more people do put octopus and calamari on the menu, this can put real pressure on these populations. And so again, understanding the basis of how sex is determined, how reproduction happens, can really lead to some beneficial advances there. I think there's also conceptual outcomes to weigh. So, and I've been hinting at this or talking about this, but the genetic or the sort of evolutionary basis of sex determination and the genetic underpinnings of it is really wild. It's complex and it's volatile. It's not sort of this immutable thing that you sort of evolve once and the work is done. And that probably relates to a number of things, getting genetic material into the next generation is one of the strongest, topics of natural sort of pillars of natural selection, right? That's the whole kind of point of evolutionary biology is how does genetic material persist? And so because of that, there's a lot of sort of, genetic action or genetic conflicts that arise sort of around this. So things like male competition, sperm competition, mother versus father, investment in offspring. There's all kinds of, overlapping, but also sort of conflicting biological motivations here that arise. And so it's kind of not through that lens, I would say it's not so surprising that there's all kinds of volatility or genetic changes. If you're just randomly sort of testing out new systems or different systems, if they work, that is, they outcompete, meaning there's more offspring that come from those, that's sort of what takes over. And so, I think I'll probably kind of wrap on this with the idea that this is not an immutable trait. And so but that's not what we're hearing from our political leaders at the moment, unfortunately. And it's, sort of, as we move into human sexuality, I'd say things get even more complicated because we're now going all the way from sort of the genetic underpinning. So the X and Y chromosome, sex determination, the SYR region on the Y chromosome, all of the biology underlying that, but all of the variation there as well. And so when that gets, I think, processed through I don't know what the right wording is here, conservative lens, if that's right, or something like that, things really sort of disconnect from the biological reality to how we as humans identify and how we sort of live our lives. And so, hopefully what evolution teaches us is that there's a lot going on under the hood, a lot we don't understand, and maybe to approach this with a little more humility and dignity for people who are living their lives would be the least I think we could ask of each other along these lines. Instead, we're seeing the opposite, as we've been alluding to, sort of sprinkling in throughout the podcast, using kind of bullying tactics attacking transgender LGBTQ plus to try to dismantle, or sort of under the guise of DEI, to dismantle our scientific enterprise. Wow, what a mess.
That in Texas, they're considering if you identify as a transgender, it's a felony.
Yeah. And so, evolutionary biology or genetics, I think, could teach you right away that this is sort of a real misreading of the situation here. It's just totally not comporting with reality, and is obviously about other things out there. Because
the president has said biology tells us there are men and women, and he's wrong. He's just lying.
Yeah.
Or maybe that's what he thinks. But you know what? What he thinks, totally uninformed opinion, has no right to inform the entire country. Zero. Nobody can stand up to him. Ashamed of yourselves. Yeah. By the way, Nels.
Yeah.
A couple of years ago on TWiEVO, we did a paper where they dated the earliest retrovirus to 480 million years ago, right? By genomic methods. And so I went and looked up what was around then. It seems to have arisen in the waters. And there's a nautiloid. Let me show you this. A nautiloid called Orthoceras, which was around 480 million years ago. And that's what it looked like. So it's extinct now. And, I don't know if the retroviruses arose in it, but it's an interesting. I'm sure that, as everyone knows, cephalopods have viruses.
Yeah.
Not many people study them, as far as I know, but they certainly have viruses.
No, I agree. I love this. And so I probably, should have mentioned or made the assumption. So how is it that we're saying, that this happened 500,000 years ago, or 500 million years ago, plus or minus, and it's because of the fossil record. So image you just showed. And then with a combination of carbon dating, other methods, that's where the sort of, kind of molecular clock or dating clock comes from. And there's also, genetic ways to do that as well. Sort of inferring mutation rates and then working backwards and then sort of comparing those things to the fossil record as well. Yeah, but your point about interesting virology or interesting biology happening underwater, I think we underappreciate this in general. So like the major pillars of our own immune systems, so VDJ recombination, this underlies somatic hypermutation. How do we make antibodies that can recognize viruses and other infectious microbes in real time? This evolved underwater in our ancestors before our predecessors came out of the sea. This was happening in things that look more like fish than humans. Same for a lot of innate immunity. So all of the hundreds of genes that we turn on in response to an infection, the so-called interferon stimulated genes, interferon signaling, that alarm bell, all also evolved in our ancestors underwater. All the major classes of viruses, Eddie Holmes and others, they troll through vast collections of metagenomic sequencing data and pull out all of these virus sequences from fish and other critters. You find all the major classes of RNA viruses. Influenza virus either evolved underwater or has moved back and forth from the air-water interface. Every major polio virus, its cousins, the picornaviruses, you find among fish. Pox viruses, you can kind of keep going down the list. All of the major classes are there. Nidoviruses that are related to coronaviruses. And so, when we think about the evolutionary history of sort of infection biology, it was aquatic. But we kind of barely scratch on that. We've really paid the most attention in this space, both in immunology and I'd say in virus biology as well, to the mammals, from kind of that interval of mice to humans, as opposed to from fish to humans. And so a lot to learn under the surface of the water.
We a couple of people have joined us.
Great.
Christopher's in Scotland cooking dinner.
Glad to have you along.
Hope you enjoy it. Christopher's in Montreal where it's snowing. Oh my gosh. Veronika Privyet from Moscow where it's 5 degrees C.
Welcome.
Welcome all of you. Okay, so and then we have some questions too.
Oh, good. Yeah, let's do that.
Let's go back to those.
Doing some live stream question filtering in real time.
Here, Les says, welcome, Les, our moderator. The Spanish royal family is a good example of inbreeding.
Yeah, inbreeding depression is a thing.
And Pete says, Queen Victoria passed hemophilia gene down to 3 of her 9 children and into other European royal families. Vanity says pollutants can alter sex as well, correct? Is it aromatase that is affected by atrazine and alters sex?
Yeah, good. I think the answer is yes. I'm not going to be able to do justice to the details, but there's definitely things that affect hormonal signaling is very much involved in or influencing sex determination. So a lot of genetic factors, a lot of environmental factors, and sort of the intermix of them that have an impact on traits related to sex determination and sort of related biology. No question about it. Yeah.
Tom's answers says that Tyrone Hayes work with frogs. It reduces male-associated characteristics.
Yep.
And Pete says we have a friend with XXY Klinefelter syndrome.
Yes. Yeah. And so the XY or XX karyotype is obviously the most common in humans, but it's not. There's many cases of different karyotypes. XXY, known as Klinefelter, XO, what we mentioned before, Turner, and a number of other sort of configurations that are out there. I'd say we're a little bit under, again, sort of in our infancy of understanding the consequences of major kind of chromosomal level variation. So in addition to sort of extra chromosomes or lack of chromosomes, there's also just a lot of aneuploidy events. So copy number, our genomes are littered with all these regions of like repeats, tandem repeats of different sizes. And even so, kind of speaking of pedigrees, I was just at a talk yesterday with similar kind of high resolution genomic analysis. And even over the course of, 1, 2, 3 generations, the number of differences, not just sort of at the letter level, the single nucleotide variants that have sort of dominated genetics, just because we can genotype those easily with the earlier technology. But as we're just glimpsing now at the bigger chromosomal kind of arrangements or sort of configurations, all kinds of bigger structural events or structural variation, as it's sometimes called. And we're just kind of, glimpsing at what that all means, or how that also influences our biology. It's going to be just as impactful or more so than the single nucleotide changes that we see.
Yep.
Yeah.
Tia wants to know about the SRY gene relocating sometimes to X, and students are curious about translocations. Can you talk about that?
Yeah, again, a great question. I'm not going to be able to do it justice since I don't follow it closely, but that's kind of what I'm trying to get at with that point about this being really volatile evolution. It's sort of like, this is not immutable. These things are changing constantly. So, a lot of the biology here as it relates to humans is in other species that have so homogametic sex determination. So 2 identical chromosomes, you get into an issue here of what's called dosage compensation, or controlling the dose of the genes. So if you take a male human that's XY, with one X chromosome, in addition to all the sex determining genes or genes involved in sex determination, there's just a lot of other genes. And if you have 2 copies, then either as a male, you need to turn up the dosage if you only have one X, or if you're a female, you need to turn down the dosage since you have 2, if it makes more sense to have 1. So there's this kind of genetic conversation that's been going on with X inactivation is one of the concepts around this. So it turns out for humans, mostly it's a story of turning down the dosage of the second X, but that's not just on one X. It sort of jumps between both. As Tia is pointing out here, the SRY might be Regis as a gene that is responsible on the Y chromosome that's responsible for a lot of male associated sex characteristics, sex traits, is not just sort of quietly hanging out in the populations. There's all of this underlying sort of genetic motion happening. And with kind of semi-randomly or randomly, and then with natural selection acting on that. So what works? And putting that together, that link, as you sort of relate that to human behavior, becomes really complicated in a hurry. And so I think we're just figuring that out, or beginning to sort of grapple with that as the technology gets better to sort of be able to see these events or to quantify them. And then the real work for the generations ahead is then to take that new information and figure out, well, what's going on here? And then through that sort of evolutionary lens, by looking backwards, can we make predictions about sort of the patterns of this and then what might happen into the future? But yeah, so translocation, this is when, one of these structural variation events where a piece of a chromosome ends up hooked on to another chromosome, that would be a translocation. And if that would happen like from Y to X, then that could be underlying what you're describing, Tia. But I have to confess, I'm not sort of, that's above my pay grade. I don't know the details of that, but really interesting biology.
Here's another one for you. Pete wants to know if gene location explains the switch from reptilian sex determination to mammalian pattern.
Yeah, good question. I think hearkening back to TWiEVO 35, strawberry sex chromosomes forever. That shows that that's possible. The fact that the genes encoding sex determination were moving around, and the sort of implication of that as well is that as some of those genes are moving through the genome, depending on where they land, does that bring in sort of the function of a nearby gene? So now if they share co-regulation or somehow, again, this is sort of plugging and playing in genetics, where a lot of these things are moving around. And then I think absolutely, if there's, again, as I'm kind of speaking at not at a sort of detailed level here, given that this isn't sort of my day job, but those possibilities exist. And so that's sort of all these genetic experiments. Then the question becomes, is in any of these arrangements, can it persist in the whole, sort of competition for getting genetic material into the next generation? If the answer is yes, then that could lead to these sort of massive differences. Now, what used to be your old sex chromosome is no longer, and one of your autosomes becomes a new one. I think I would point to, I don't know about the reptilian systems, which are a bit less studied, but probably the best work here right now in terms of having the tools and resources and some of the observations is in some of the Drosophila systems. So Drosophila melanogaster is sort of the workhorse of the invertebrate fly model or fruit fly model, but there's other closely related species where some of this action has been happening. And there's several really great labs that are kind of on the case or working on this.
So Tom says, but aren't these human SRY translocations very rare, and DSDs can be sorted into male and female developmental pathway defects?
Absolutely. And so, I think it's here maybe and evolution is sort of marked by all of these rare events, and then that are constantly happening that we generally, very rarely see, if we see at all. And so that's kind of one of the fun pursuits of the field is to try to make the rare more obvious, or to weigh that in ways that might not otherwise be obvious. But then absolutely, so then selection acts, and so I would say in a lot of these cases, so I don't so let's just take Turner syndrome as an example in humans, the XO situation. So that's also one of the traits with that commonly is infertility, and so in that case, it'd be very unlikely that selection is going to act for, basically a new sort of system of sex determination, because we see those appearances somewhat rarely, although not as rare as you might think. And then it doesn't have that sort of beneficial outcome of outcompeting. And so that's most of evolution. That's business as usual. It's the rare cases where something sort of then has the benefit or the sort of competitive even advantage, where then you can see these turnovers. And so, I don't know of cases like that sort of in mammalian lineages, but it's certainly happening. And as we kind of glimpse at a lot of the diversity of life around us, like the tetrahymena mating type system with 7 mating types, you can see that there's a lot of room for evolution to get pretty adventurous in what sex determination looks like.
Want to do some picks, Nels?
Yeah, more than anything, let's do that. So our science picks of the week, I'm going to start. I've got one came across my radar, sort of an occupational hazard here. Speaking into a microphone. This is a story on NPR that picks up a recent paper in the Proceedings of National Academy of Science. Your audio quality can impact the way people view you. High quality audio can make the speaker seem more attractive and convincing. I don't know, maybe we've done the experiment over the years, Vincent, right? When as I've slowly followed your lead to try to upgrade my audio, if I sound like I'm in a tin can, that probably has some negative impacts. This also extends sort of in our Zoom. So the researchers here, it's a fascinating study. They did, some kind of social science experiments where they gave sort of the same scripts to people kind of over Zoom scenarios. I think they were doing job interviews or something like that, and then asking people to rate the candidate. And it turns out, maybe not too surprisingly, but your microphone, how good your microphone is, influences how your audience sort of receives you in many different situations. But wouldn't you say, Vincent, you've kind of early and often for Microbe TV, you've sort of invested in good audio, good video. That's sort of one of the pillars here of
well,
what you do.
we started as audio only, right? With
Right.
TWiEVO and TWiP and TWIM, and it wasn't until 2020 really that pandemic time when we started using Zoom and included video. So for many years, just audio. So I thought this is all people are getting, so it better be really good. So I invested in good microphones, and I sent microphones to my hosts because I didn't want them using crappy mics in their laptops or whatever. I always felt that the audio is an important component, even in video. People are influenced by the audio, although less so because they have the video as a crutch, right? So they're kind of more focused on that. But I always make high quality video. We've always edited it to sound really good.
Yeah.
It really kills me when a host, if we have some remote host who's new, has horrible sound, right? It's like, it's hard to listen to and get through to what you're supposed to be doing, which is learning. So Yeah, to this day, we think this. So this is not surprising to me.
Yeah, exactly. I agree. No, it's great. And they point out too, like the video, we get immediate feedback. Like when we're on Zoom, we can see ourselves. We probably stare at ourselves for hours now, more so than it's like looking in a mirror. But we don't really have a sense of how good our audio is. That's what comes across to the audience, but not to us. We sound like we usually sound, but how that gets received is different. And you're right. I think, in the past when we've had guests on, if they don't have a good audio system, that sort of difference in quality really makes it hard to focus on the conversation. It really just kind of can kind of get in the way. So anyway.
So I just want to point out that the audio on Zoom sucks.
Yeah.
Totally sucks. Mm-hmm. So it's very hard to improve it. All right. And so when we started doing Zoom during the pandemic, I didn't like it at all. And I looked for other alternatives, and this Riverside, which we have settled on now after a few other tries, gives you very good audio because it's recorded locally. So as long as you have a good mic, it'll sound good. But I think Zoom is horrible. And they do that because they want to compress it a lot to make it easy for a lot of people. I mean, they're very good at having hundreds of people on a Zoom call. That's great if that's what you need. But the sound in the video is horrible.
Yeah. No, I guess thanks to, is it Ray Ortega who's helped over the years with kind of...
Yeah, so early on, I hooked up with Ray at ASM, American Society for Microbiology. He taught me a lot about audio, and I've also tried to learn on my own what makes good. I mean, watched lots of YouTube videos. I read a lot of things. I bought books to try and figure out how to make good audio. And I used to do all the audio for all the podcasts myself. And a couple of years ago, I think during the pandemic, I hired someone, professional audio engineer, sounded worse than mine.
That's right.
So after a while, I said, I don't need you anymore. I'm sorry, you're too expensive. But
there you go.
The thing is, you get to do it a certain way, and then someone else does it differently, and they don't pay as much attention. But there's certain things that makes audio clean. One is that you don't want background noise, right? Every room has some kind of background noise. There are ways to take that out, but you have to be aware of it. And you have to have a nice frequency distribution. There are certain frequencies in human voice that are disturbing, and you can pull those out. So all of this makes for a nice sounding audio. And if you just have audio, that's key. So, and the author of this, by the way, article, Nell.
From NPR.
Something Boyce.
Yep.
I talked to her.
Greenfield Boyce. Nell Greenfield
Boyce. Yeah, I talked to her years ago. I don't even remember about what, but it's been around for a while.
Yep, exactly. All right. Well, that covers kind of, TWiEVO under the hood here. We actually do really, and Vincent in particular really thinks about the audio, which helps with our, think about how bad we would sound if we didn't have good audio, or how less convincing we would be.
I mean, this Livestream program, which we use, is called eCam. It's okay. It gives a pretty good audio. It actually records our tracks separately. And so I make a separate audio-only podcast for people that want to listen to it. And I think it sounds pretty good.
That's great. Yep. And it allows us to do this in real time and to interaction in real time, which also has some massive advantages. How about you, Vincent? What's your science pick of the week?
I could pick for so many things, right? What's going on this week. But my pick this week is a New York Times article. When RFK Jr. was asked about the measles outbreak a couple of weeks ago, he said, take vitamin A and cod liver oil. So now this article is reporting that parents in West Texas are giving their children high doses. They think it prevents infection, and these kids are showing up in the emergency room with liver damage.
Oh, my God.
So this is not surprising. Vitamin A is a fat-soluble vitamin. So it's going to concentrate in organs like liver, and it's going to cause damage. Now, so what does it mean about vitamin A? So it turns out in resource-poor countries where many kids are malnourished, a dose of vitamin A will make measles less pathogenic. So you will have less. So for kids who are malnourished, measles is 300 times more lethal than in other countries. So in those settings, vitamin A is useful. It's not useful at all in the U.S., where people are properly nourished. And it certainly does not prevent infection, which, these people don't know. He says, take vitamin A. He doesn't even give them any guidance. No. So they just inject their kids or give their kids it, and they're getting sick. This is horrible, horrible, horrible. He should be fired and sued for medical malpractice, even though he doesn't have a
medical license. Yeah, correct. Awful. This is good reporting by Teddy Rosenbluth, and important to get this word out here because, this is, to me, just another example of this disconnect between confidence and competence. We hear from RFK Jr. confidently saying, "Oh, just take vitamin A or cod liver oil," but as this lays bare, the consequences of that for public health is awful. We're going in the wrong direction, and it's not. This isn't sort of rocket science, right? This is pretty straightforward stuff. But so either there's, a lack of curiosity about how things really work, or there's nefarious things underneath the surface. I saw, you probably saw this too, whatever RFK Jr.'s sort of false nonprofit for public health, the child health thing or whatever, cribbed the CDC website and put on this kind of, information as if it was coming from the CDC. So it's kind of happening on two levels, and continues to do so with the conflicts of interest that are still sort of front and center here. I think RFK Jr. said he wasn't going to profit from the lawsuits on vaccine makers, but his son will. And then, that's 1 degree of separation and with behavior like this really doesn't take away from the increasingly sad impacts as the measles are gaining steam. I think I was reading yesterday too, that there's cases now in DC, or a case in DC that might be spreading. We're north of into the several hundreds now in Texas and Oklahoma. I think the estimates, again, this is outside my wheelhouse, but the estimates are that this outbreak will be going for at least the next year on its current sort of burn.
So this started in a Texas community that is historically under-vaccinated. The Mennonites homeschool, so they don't go through any school vaccination requirements. What should have happened at the very outset is HHS gets up and said, we need to vaccinate everybody immediately, go in there and vaccinate people. I'm sure they would have allowed that in the context of an outbreak. But no, he said, take vitamin A and cod liver oil. I mean, this is medical malpractice, in my opinion. But he just doesn't have a license, so we can't. So there's a great quote in this article about, so Kevin Griffiths wrote in the Washington Post, apparently. Until last week, he was communication directors at CDC. He resigned because of Kennedy's handling of the outbreak. He wrote, in my final weeks, I watched as career infectious disease experts were tasked with spending hours searching medical literature in vain for data to support Kennedy's preferred treatments. Yeah. This is such nonsense, folks.
Yeah, backwards. And this is that kind of pretending, like we're pretending to do science when you when you flip the script and say, well, here's what I think, or here's my agenda, and let's find evidence for that. That's what's happening, presumably, with the whole kind of trying to reestablish a false link to vaccines and autism. Good Washington Post opinion piece from a public health reporter, a fellow named Donald McNeil Jr., kind of a little personal contact. His brother is one of my neighbors here in Salt Lake. But he was calling out RFK Jr. for, I think what he called libertarian childishness. And so, in pretty blunt terms, this is worth looking up, but if you haven't seen it already, but in pretty blunt terms, how do we think about public health, like, and things that where it's compulsory? So seatbelts, how have a society have we or smoking cigarettes on an airplane? We've kind of crossed the threshold on those ones where we're pretty comfortable with there being seatbelt laws in place. We're pretty comfortable.
Speed limits too, right?
Speed limits, no smoking on an airplane. we've kind of come around to that, but somehow we've let go of our
So that was a great opinion piece you're talking about. That was by Don McNeil.
yeah, Donald McNeil Jr.
He
Exactly.
said there's certain things you give up in a nation, and he said that's what makes to protect each other. That's what makes us a nation, and not 340 million hunter gatherers only concerned about ourselves.
Yeah, that was a good
Perfect.
line. Yeah,
This personal freedom idea is just nonsense, folks.
yeah,
You
no,
don't have the freedom to infect other people. It's not right.
yeah.
By the way, Tryon says, are there any monoclonals against measles? And Christopher Robinson said, I don't know of any. That's correct. I don't know of any. There are no antivirals. They're just vaccines.
Yeah, and so that's another kind of piece of this puzzle, right? So what did we? So let's say that you do want to get away from vaccines, and counter viruses. So one of the things you could do is invest in a lot of research to come up with new antivirals, whether it's measles, whether it's dengue, West Nile virus, yellow fever virus. Speaking of Texas, these are probably pandemics that could be on the menu in the future as well. And so what did we see yesterday from Health and Human Services, NIH, canceling an entire program that was devoted to exactly this, to find new antiviral treatments sort of across the board.
Yep.
And so it's sort of, this is kind of that confidence versus competence, the double hit here of, well, we don't want to do vaccines, so take cod liver oil or vitamin A, but we also don't want to invest or understand what we're investing in, and we're just sort of cutting under the sort of false umbrella of efficiency. I would love it if we had great new antivirals that meant personally, if I didn't have to take a get a flu vaccine shot every year because we had a different sort of intervention, count me in. I'd love to do that research. I'd love to team up. And currently, there's a lot of scientists who are finding themselves with a lot of uncertainty about whether that will even be possible. But all these things are underway and on the menu and are threatened right now. And so it's sort of kind of, incompetence at both ends of the pipeline here, I would say.
So Les sends a link to another New York Times article, the U.S. to end vaccine funds for poor countries. Yeah, this is part of the USAID cutback. So this is just mean, right? There are a few countries in the world that can help others, and the U.S. has always been one of them. And now, for some reason, because the people in charge don't like it, they're backing off. And I think this is horrible. It's a terrible precedent. A lot of people are going to die as a consequence, but they don't care. They only care about themselves. But I always thought what made America great was that most Americans cared about other people. And I always thought we should take care of the rest of the world with medicines because we can. And I just do not understand why you have to shut it off. The amount of money involved is really nothing. It's unfortunate.
Yeah. Yep. A lot to unpack there, but it's, really, as we look at the directions, like in all directions, I think, similar themes coming up sort of across the board with the sort of chaos, recklessness, sloppiness that's going into this, which is really leading to a lot of tipping points for a lot of programs from health, social security, national security, sort of place in the world. I mean, the U.S. obviously has a checkered history to say the least, but in places where we're putting resources to try to do better, there's something bigger that really is under attack in many ways, shapes, and forms.
So folks, if you're in some of these states with special elections, please flip that House, flip Senate, please. Otherwise, this is going to kill us. Literally.
Yeah, that's right. Some rays of hope there. I think I just saw last night there was a special election for maybe the state Senate in Pennsylvania. I think the Senate, the House, state House, and the governor are now all flipped in Pennsylvania. And so hopefully that's sort of a hint of things to come. Yep. There's a big, a really great example of sort of putting the thumbs on the scale as it relates to Elon Musk, the Supreme Court situation up in Wisconsin right now. I think we'll all be watching that closely as money and sort of shenanigans are being pumped into that situation. That'll be a sort of stress test for where we are in our political system right now, too.
All right, let's wrap this up, Nels.
Sounds good, Vincent.
I want to thank our moderators for today, Barb Mack, Vanity Nutrition, Tom Steinberg, and Les. Thank you all for joining us and moderating, which we always need. Keep it civil. Thank you. And thanks all of you for coming. We had about 45 of you and 32 likes. Hit the like button in these last minutes to show people where the real science is. You can find TWiEVO at microbe.tv/TWiEVO. There is an audio-only version there. I'm a little behind, but you can go there for that. Mm-hmm. And we would love to have your support if you like these programs. Microbe.tv/contribute. Nels Eldy is at cellvolution.org and Early Bird on Blue Sky. Thank you, Nels.
Yeah, thank you, Vincent. Good to be together. We'll be back next month as we continue to weather this Category 4 hurricane of all attacks on science and in different ways, shapes, and forms. We're trying to keep an eye on it, give you our impressions, and we'll keep doing that as we continue to share the science that has been so generously underwritten by all of us, the taxpayers, and has really kind of points to how science is a crown jewel in this country and how this could be going away. And so let's all continue in this conversation. Continue to love feedback, ideas, also your sightings out there as well. Share this with us early and often. Are there things that should be on our radar that aren't, or are you seeing other opportunities or things like that? Let's go through this. Let's walk through this together. Thank you, Vincent. Good to be together. I'm looking forward to the next one.
I'm Vincent Racaniello. You can find me at microbe.tv. Music you hear on TWiEVO, let's turn it on, it's by Trampled by Turtles. You can find their work at trampledbyturtles.com. Been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious. Bye-bye.